Questions is yours. You are muted, Duncan.
Correct, I'm muted. Okay, let's get this going. So hopefully you're seeing my presentation screen.
Yes, not in the presentation mode.
Yes. Okay. What about now?
Yes, now it's working.
Yeah, fine. Okay, good. So yeah, I'll start by saying thanks very much for the nice introduction and for the invitation to come and give a talk. So what I'll talk about now is some of the work from the group over the last years where we have used nanoscale engineering to control how cells can interact with proteins and form adhesions. So it's sort of something between material science and life science. So I would point out that we are in Denmark, at least I am, and Sarges as well. So we're in Aarhus University, which is here on the mainland of Denmark, second largest city in Denmark. So what I want to talk about is the formation of tissue and proteins and cells, and I want to talk about length scales. And so for me, the nano is the important thing, the nanoscale. And what I would point out is that tissue is organized on multiple different length scales. And over the last decades, we've formed the ability to do engineering where we can create structures on those different length scales as well. And so they can provide us with tools that can allow us to study cells and proteins and tissue at different levels. And so the work I'm describing today is where we are working with nanoscale patterns, sort of between 100 nanometers and a micron, and we're using them to study cells which are in the tens of micron range. So one of the long-term goals of this work and of the group is to be able to understand how cells respond to their surroundings. So here's a sort of schematic picture of how a cell might be situated in vivo. It'll have signals coming from the extracellular matrix. There'll be signals coming from cells in the surroundings, either by cell-cell contacts, or there'll be soluble molecules either from elsewhere in the body or from local cells. And so this sort of set of signals will help the cell to decide what to do, how to respond, this combination of these extrinsic factors with its intrinsic factors. And for us, that would be, it's interesting to study, but it would also be interesting if we can use that knowledge in order to control, for example, how stem cells will differentiate and one way to do that could be by making more advanced material surfaces which can provide some of the correct signals from the surface and then give an appropriate response from the cell and steering of the cell's behavior. So what I'll talk about today is where we've tried to use nano patterns of proteins in order to study and control the formation of different adhesion types within cells. And so here's another schematic picture. This is a cell which is in contact with neighboring cells and also with the extracellular matrix. And there's a number of different types of adhesions that the cells can form. It uses different cell membrane proteins. And so there are integrin-based adhesions which will form and they'll sort of connect with the extracellular matrix. There's also a number of different cell-cell contacts. And so our approach is to mimic the thing that the cell is interacting with, making a pattern of the appropriate proteins, which could be extracellular matrix proteins, or they could be membrane proteins from the surrounding cells, and use that to study the formation. And we have this hypothesis is that the size of things matter and the pattern matters, not just the specific interactions. And so this size scale in itself can give you some specificity, and that'll give us a possibility to control how cells will respond, adherent cells. And the idea being that if we create a pattern of proteins at a material surface, we will use that to control organization of proteins within the membrane of cells in contact, and then that will give you the possibility to give signaling events. So we start a little bit about the engineering. There's a whole range of different techniques one could use. We've extensively used a particular technique called colloidal lithography, and my computer is now crashing. Oh no, now it's back. Yeah, so we've extensively used a technique called colloidal lithography, which is Yeah, really, my computer's not working anymore. You still hear me?
Yes.
Yes.
I don't know what my computer's doing right now. I am going to stop sharing and try again. Oh, maybe it's back. Okay. Yes. So we've extensively used this technique called colloidal lithography, and it's good for the range from maybe 100 nanometers up to a few microns in size. It's the advantage of being a self-assembly bottom-up technique, which means you can create pretty large areas at once, and that's useful for the cell culture experiments. And now my computer's going slow again. Okay, so the idea with the colloidal lithography is that we use colloidal particles, we absorb them onto a surface, and then we make a pattern of those particles, and we use that as a mask, and then we can transfer the pattern of that individual particles so that one particle will allow us to create one structure on the surface. And so you can use that to create topographies and shapes. You can do some optics and other types of studies, or you can use it to create chemical patterns. And that's what we've used to do these protein patterning studies, is using this colloidal approach to make chemical patterns. So we stick the particles onto the surface. Those particles will arrange themselves on the surface to do with random sequential absorption. It's sort of well understood physical process, and it leads to short range ordered arrays where there's no corresponding with position from short distances and far away. But there's a characteristic spacing which we can control through control of the electrostatics. So we have a pattern which has a short range order. And this is a technique that we've been using over the last decades. We can make a whole bunch of different structures, and we use it in different fields. And so it's really a sort of a broad technique. So in terms of looking at cell surface interaction, the work that I'll describe today actually started out from working with studying the effect of nanotopography on cells and not nanopatterns. And so at that time, a little over 15 years ago, we had this idea that nanoscale topography on the surface of medical implants could have some effect and control of cells in the surroundings. And that's definitely turned out to be the case. This is looking at fibroblasts on polymers, which were either very flat or they had 100 nanometer pillars. And here on the flat surfaces, the cells can form mechanical contacts. They can have a well-organized actin skeleton. And when you have them on these pillared surfaces, it sort of really interferes with that. And so it's something that we saw with different cell types on different materials. So it was really sort of a rather a broad response, but rather specific also. And then we would find changes in for example, signaling events with the cells. If we're looking at stem cells, then we saw changes in differentiation of the stem cells. And so it's really promising. It's a little hard to understand exactly what's going on. And so one of the hypotheses we had was that the role of the topography was that it would change the way in which proteins were available at the surface. So as we all know, if you have a material surface and you interact that with cells, then that material surface will get covered with proteins, and those proteins will allow the cell to have specific interactions with either proteins from serum or specific proteins if you put them down yourself, or if the cell is remodeling its environment. And so we had this idea that this is a sort of fibroblast on 100 nanometer polymer pillars, and it was very clear that the cells were not able to reach all parts of the surface. And so we had this idea that the role of the nanoscale topography was that it limited the cell's ability to interact with surface proteins, and that would change the way in which it could form adhesions. And so that on a flat surface, it could have very large adhesions, and on a topography structured surface, then it would have smaller adhesions. And so that was actually for us a hypothesis. And then we went into this protein patterning in order to test that hypothesis. And then actually the protein patterning has turned out to be really interesting, and we've been running with that ever since. So the idea of having a protein pattern is that you predefine a pattern of a particular protein. We do that by first generating a material with a chemical contrast. And so you'll have something like, using the colloidal lithography, you can have these, this is a 300 nanometer particle, will give us a 300 nanometer region with one chemistry and another chemistry. We then want to convert that into a pattern of proteins by controlling where proteins can adhere and how they adhere to the surface. And then we can use that to study how cellular complexes form, adhesion complexes. And so one thing I wanted to be clear about and point out is that these nanoscale structures are much, much smaller than cells. And so we're not looking at controlling the overall cell shape. We're looking at controlling the formation of complexes and assemblies much smaller than the cellular size. This colloidal lithography approach, we have used it to create chemical patterns of gold and silicon dioxide. This has turned out to be a pretty robust method, and we've been using it for at least a decade now. The idea with that is that here we'll have gold in the circular patches, which will be created from the nanoparticle size, so we can vary the size of that from, say, 60 nanometers up to several microns. We choose to make that gold having a hydrophobic character by using alkane thiols. And then we're able to assemble a PEG-containing block copolymer, which will then adhere strongly to the silicon dioxide part, very weakly to the gold, which has been made hydrophobic. And so then we can have that the surface has a protein rejecting character, except where these circular regions are. And then we can just bring in extracellular matrix proteins, fibronectins, vitronectin, allow them to adhere, and they'll stick to these hydrophobic patches, and then they'll form us a pattern. And so there's an example here, SEM images of different sizes of patterns from 100 nanometers up to, whatever that is, 3 microns. And then these are corresponding fluorescence images using antibodies to stain for particular proteins that we put there. This is a fibronectin pattern, and this is a vitronectin pattern. And then we're using those patterns. So these are sort of a range of pattern sizes to interact with cells. And again, just want to stress that the patches of protein that we're creating are much smaller than the size of cells. So it's not like micro patterning where you would be controlling the overall cell shape. So we've been working for this for a while. We are able to pattern different extracellular matrix proteins. So we've worked with a range of those. We also have been working with cell membrane proteins, and you don't have the full membrane protein, but you take the ectodomain and orient it on the surface. And then also just to point out that we would typically use something like serum albumin as a control. So we would have patterns of the same type, but with albumin. Yeah, so that's a patterning approach that we have. It's not perfect, but it's robust. It allows us to create tens of square centimeter of sample. We can do experiments with millions of cells. We can look at cell populations. And so now I'll talk about some different studies we've done to look at how cells are able to form different types of adhesions. And so the first one that I'll talk about is this thing, focal contact or focal adhesion. That is an adhesion between the cell, integrins in the membrane of the cell, and the extracellular matrix. And then it links up on the inside to the actin cytoskeleton. So it's a mechanical contact between the extracellular matrix and the actin skeleton. So these focal complexes and focal adhesions are involved in the cell being able to exert force. And so it's very much involved in motility of cells. And so if you have a cell which is traveling, the schematic picture, of course, traveling in this direction, the green here is meant to represent actin filaments, and the red is meant to represent these adhesive contacts. And so when the adhesive contacts are first forming, they're typically called focal contacts. They're very small. They're sort of 30, 50, 100 nanometers in size. They form at the leading edge. They are then maturing, becoming larger, and then they're typically called focal adhesions. And then they can connect up to actin filaments. And then the cell can exert force through the actin filament onto the adhesion, and then sort of pull itself forward. So this is something that we were studying. This was in cell line C2C12s. And here we wanted to create different patch sizes of protein which covered the range from these focal complexes, which are down at sort of 50, 100 nanometers in size, up to the several micron size focal adhesions that you will typically see in cell culture. And so we can do that, and we can create protein patterns. And then we did that with different protein types. So we have one line here, which is for fibronectin patterns, and one for vitronectin patterns. So they're two different extracellular matrix proteins. They bind to different integrins within the cell membrane. And these are the smallest patterns, 100 nanometers up to 500 nanometers, and then from 600 up to homogeneous. And so the first thing that we see clearly is that we have a really drastic effect on the cells. On very small patterns, they are not really able to form strong contacts. They cannot have a well-organized cytoskeleton. As the patch sizes get larger, they're able to form larger adhesions and to spread. And so you can sort of immediately see there are large effects. If we look close up, we can stain for some of the proteins which are in this focal adhesion. So a focal adhesion It's formed with an integrin in the membrane of the cell, linking to extracellular matrix proteins. And then there are a number of different adapter proteins which will be recruited to the focal adhesional complex, which will then link up to this actin skeleton. And so if we look for some of these adapter proteins, this is looking for vinculin, we can see that the formation of these focal adhesions is very much directed by our patterns. So we have patches, they're forming above the patches, are the same size as the patches, and so we're able to control the formation of these focal adhesions. So sort of the first message here is that we can use this pattern that we've created in order to control the formation of these focal adhesions, and then that actually has a significant effect on cells, such as spreading. We can use this as a tool to study the formation of these focal adhesions. We can look at different components in this focal adhesion. We can look, for example, here we're looking at a cell fibroblast on top of another C2C12s on top of a vitronectin pattern, 500 nanometer pattern. And here we're looking for the alpha V, which is one of the two subunits in the integrin, which will bind to vitronectin. And so we see that those are forming above the patches. They're the same, controlled by the size of the patch. We can then look at different proteins which are in this sort of recruitment. I showed here this for looking for vinculin. We've looked also for zysin and paxillin and a number of other proteins. And one thing which surprised us at the time, and it seems to be a fact, is that it's not the size of the focal adhesion that you're forming doesn't really correlate to the maturity of the focal adhesion in terms of which proteins are recruited. And so the formation or the lack of formation of this connection to actin is not that these proteins are not present. It's got to be something else. So what we did see, though, was that so there's a threshold behavior for cells in terms of when they can form adhesions. And that seems to be dependent upon which integrins are involved, in that it depends on which proteins that we put down. And so this for the C2C12s, we find that on a vitronectin pattern, you can only form once you have a patch of up to 200 nanometers or above. But you need much larger patches for fibronectin patterns. And so that's something that we consistently see. It depends on which cell type, and I'll get back to that later on. We also see is that we can control the formation of these little focal adhesions. So this is looking at vinculin again. This is in a mesenchymal stem cells on vitronectin patterns. So these are slightly larger patterns, 1 micron patterns. And you can see clearly these little focal adhesions forming with these staining for vinculin. But what's clear is that the cell can link up several of those focal adhesions to the same actin filament. So we're not actually controlling the number of adhesions that of focal adhesions that the cell can integrate into a fiber. What we are actually though controlling is the width of the fiber that can form. So this is on these are the mesenchymal stem cells again. Looking, the red is the actin and the green is the vinculin forming these focal adhesions. And so this is on a homogeneous surface. And then if we go from larger patterns down to smaller patterns, and the size of these fibers that form is getting less and less. And so it seems to be the width of the pattern is the key parameter in terms of having control, and it's the size of the fiber that the cell is able to form that is the key parameter. Yeah, so we have a system now for controlling the formation of these focal adhesions. And so we then want to sort of do some science with it, try to understand how that's what that's doing to the cell. And something important to know about these focal adhesions is that they're not just mechanical contacts. They do form this connection between the extracellular matrix and the actin cytoskeleton, but it's also a signaling center in terms of when the cell exerts force, different reactions go on within the focal adhesion and you can have signaling events triggered. And so this is all part of this mechanotransduction process. And what's also interesting to note is that well, we have this hypothesis that the amount of force that a cell could exert would be related to how big this adhesion was. And what's relevant here to know is that in tissue culture wells, these adhesions are typically very large. And the cytoskeleton that forms the actin filaments are very fine and well large. But that's rather different if you go into a 3D culture system. And typically these focal adhesions, focal complexes are much smaller, and it's not really clear how they form in vivo, but it's clear that there are not these large micron-sized focal adhesions forming. And so what we hoped to do was to change the size of focal adhesion that a cell could form and see if that would alter its behavior, if we could change its mechanotransduction signaling. So in this work, we turn to working with the stem cell type. So this is epidermal stem cells. And here we made patterns of fibronectin. And so then the cells forming these focal adhesions on top of the fibronectin patterns. And like with the other cell types and the other proteins, we see that the cell's ability to form these focal adhesions is controlled by the patch size. So this is a 100 nanometer pattern going up to 3 micron pattern, and then this is the homogeneous protein. And so what we see is that on this, in green, we have a vinculin, so that's these focal adhesions, and we see that the clearly size of these focal adhesions that can form is controlled by this patch size. And what's interesting here is that when we move away from these cell lines, which are sort of adapted and conditioned to tissue culture polystyrene, then the cells are able to form focal adhesions or focal complexes already at very small sizes. So at 100 nanometers, we're already seeing adhesions form. And so at the same time as we're having this control of the focal adhesions, we see correspondingly that the cell's ability to link up to those focal adhesions and to form actin cytoskeleton is also altered. So with these epidermal stem cells, they are sitting at the basal membrane of skin, and they will differentiate into keratinocytes. And so one of them, we can look for markers for that keratinocyte differentiation. And so if we look for involucrin, that's in green here, it's a marker, and we clearly see that there's a big difference depending on the patch size. So on the homogeneous surfaces, we see little differentiation with these focal adhesions being formed. And then as we get smaller and smaller patches, we have more and more differentiation, and that's sort of quantified here. So clearly we're having a very large effect controlling the cell's differentiation based purely on the size of focal adhesion that this cell can form. So we had a hypothesis that it was to do with mechanotransduction. So we wanted to see whether or not the cells were altered in their ability to exert force. So in this work, we were able to make use of this force sensor protein VIN-TS. This was developed in the Schwartz lab in the US. So we have that, and we could recombinantly introduce it into a cell line and do some studies. And so the idea with this protein is that it's like the original vinculin, except that it's had a force sensing block in the middle, and that force sensing block has two fluorophores, which are, if they're very close together, then you can have energy transfer, and then there's a distant dependence fluorescent resonant energy transfer. And so if there is a lot of force, then these are far apart and there is very little fret. And then when they're little force, then there is a lot of fret. So it's sort of a force sensor. And so then this vinculin is present as one of these adapter proteins in the focal adhesions. So if there's force being exerted on the actin, then you could imagine that you might have a response from this force sensor. And so we then looked at patterns of different sizes. This is based on optics. So in order to get good data, we need to have large pattern sizes. So we can't look at the very small pattern sizes, but we have the 3000 nanometer patch and the 600 nanometer patch, and then we compare that to this homogeneous surface. And there are slightly different size focal adhesions on each surface, so we sort of plopped all of them. But what we see clearly is that on the homogeneous surface and on the large pattern size, and we have a low fret value, which means a high force. And then if we are looking at the smaller pattern size of 600 nanometers, and we have a much higher fret force, fret value. And so that fret signal, the fluorescent signal coming out, tells us that these fluorophores are close to each other, and so there's low force. And there's actually must be very low because it's about the same value as we get when we bring in babistatin and sort of disrupt the actin, and where there should be zero force. What we see clearly is that on these smaller patches, the cells are just not able to pull as hard. Although we'd seen before that there wasn't much difference in the sort of the recruitment of proteins to these focal adhesions, we wanted to do a sort of more thorough study here. So we looked at a whole range of different adapter proteins present in these focal adhesions, compared the intensity of those staining for those different markers, and then seeing how that changed patch size. And so we did find that for smaller patches, we had a bit higher intensities of some of them, but there was very little difference in the ratio between them. So, I mean, there's perhaps with vinculin, is another case. Vinculin is the sort of purple color here, and it does seem to be a bit lower on some of the smaller pattern sizes. But overall, it seems that the proteins which are incorporated and recruited to these focal adhesions is not affected by the patch size, but it's the amount of force that a cell can exert. So we chose to look a little bit closer into vinculin, since it was showing something a little bit different. And so there we also looked at not just was vinculin present in these focal adhesions, but what sort of binding strength did it have? And so that the way that we did that was that we would bleach use FRAP to bleach the fluorescence of the vinculin that was present in a focal adhesion, and then we would look at how long it took to recover. So that would tell us something about the dynamics of the binding and unbinding of vinculin. And so what we find there is that on the smaller patch sizes, we have a faster recovery of fluorescence, and so that corresponds to a quicker exchange, and therefore it's sort of somehow destabilized. So that could be that that's the way in which force is the cell cannot exert force because the vinculins are coming off all the time. It's something to do with the dynamics. Yeah, so but from this study, what we could see was that if we take our stem cells, we are able to control the adhesions. We are not changing which proteins are present in those adhesions, the recruitment, but we are changing the way in which a cell can exert force, and that is changing the mechanotransduction and the differentiation. What's interesting is that the differentiation pathway that we induce for these very small patch sizes is actually very similar to what we see in, or not what other people see in hydrogels. And so what we believe is that by preventing the cell from exerting force, even though it's on a rigid surface, we're sort of fooling it into thinking it's on a soft material, or at least acting like it's on a soft material. And that could be something that's then relevant for advanced tissue culture, where you could have a 2D system, but you could still have a sort of a feeling of a soft material. So I'll briefly touch on some other adhesions to extracellular matrix. So this was focal contacts and focal adhesions I've just talked about. That's connecting the cell's actin cytoskeleton to the surrounding extracellular matrix. And there's another type of adhesion which is connected to the extracellular matrix, but that connects not to the actin but to the intermediate filaments. And so in skin cells, that would be the keratins, so the name for the keratinocytes. So these are hemidesmosomes, and this is actually Sadeg's work. So this is one of his projects. So it's an ongoing project, but the idea there is to make patterns out of an appropriate protein that's involved in the formation of these hemidesmosomes, and laminin 332, or previously laminin 5, is the appropriate protein. These adhesion complexes, they are integrin based, so there's an alpha 6 beta 4 is the integrin. It binds to the laminin 332. And then it recruits a number of different adapter proteins, as it's collagen 17, pectin, dystinin, and then it connect up to the intermediate filaments, the keratins, and sort of it's sort of an analog of this with the actin, but it's another type of adhesion. And probably what's important here is that if you take a skin cell, then the times that it's going to form focal adhesions is where there's been a wound, where there's fibroactin has been placed, and then you're needing to close a gap and have a motion of the cell. When the cell is just normal and it's in its normal homeostasis, then it's binding to its extracellular matrix through these hemidesmosomes. Yeah, so here's some of Sadeg's data. I mean, he makes patterns of laminin 332. He's able to observe that the integrin alpha 6, blue one here, is forming above the patterns, and we're able to control the pattern size. And then also that we're getting recruitment of some of these adapter proteins. So this is collagen 17, the green protein here. So we're clearly having ability to control the formation of these hemidesmosomes, and so the next step will be to see how that's involved in cell signaling and cell differentiation. Yeah, okay, so those we're looking at two different adhesion types between cells and extracellular matrix. There's also a lot of interesting adhesions between cells and signaling going on, and so we've done a couple of studies where we've tried to look at mimicking or forming one of these cell-cell contacts between a cell and a nanopattern. And so the idea there would be that we would take the membrane proteins, not the whole protein, but the ectodomain, extracellular part of this protein, and form it on a surface in a pattern. And so I'll give an example of that, of forming these adherence junctions. And so there, we again are using the same idea of having the colloidal lithography giving us a chemical pattern, and using this PEG polymer surface in order to prevent proteins from binding in the background. We have to work a little bit harder in order to get the protein to be functional, in that it needs to be oriented correctly. So this ectodomain of the ECAD, ECadherin, which is involved in the adherence junction, is fused onto an FC domain from an antibody, and that FC domain will bind to protein A. And so then we have an assembly where we put down eutravadin, which is non-specifically bound to the surface. We then have biotinylated protein A, and that allows us to then assemble the ECAD in a functional form in a pattern. So here we have our patterns forming. So this is 800 nanometer pattern, 300, the limit of the resolution, but you can kind of see that there's a pattern in there. And this then we can use to study the adhesion of cells with adhesion junctions onto these surfaces. And so we have homogeneous 800, 300 down to 100. And what we clearly see in this case is that there's a threshold for adhesion somewhere between 100 and 200 nanometer patterns. So you can see the quantification here, and we've done, if you do multiple repeats, you get different numbers of cells, but you see the same result. And so if you start counting and estimating how many of these ECadherins can fit above a patch, and at the 100 nanometer patch, you can fit about 6 ECadherins, and the 200, you can fit maybe up to 17 as a maximum. And so somewhere between there is the threshold for how many ECadherins you need to be engaged in order to have an adherence junction form. But as well as the formation of these, the sort of cell attachment, we also see changes in the cell's ability to spread, connect to the actin. And so on the very small patterns, they're just not spreading at all. And then there's a sort of a size dependence in the spreading. And then we also see changes in the way in which the ECAD is assembled and how it links up to the actin. So again, we're seeing a clear connection between the patch size, the formation of these adhesions, and the connection to the cytoskeleton. And now it's waiting. Yeah, so then the sort of summary of this is that we see that you have a threshold behavior in the adhesion formation, and that you need to have up to this maybe 17 ECadherins in order to be able to get an adhesion to form. Yeah, so that's looking at ECAD and looking at adherence junctions. So we were interested to look at another cell-cell contact, and we wanted to look at something maybe that was a bit more sort of functional, something that would be giving us some insights into some process within the cell system. So this is a study where we looked at monocytes and monocytes adhering to the endothelium in sort of the vessel side, the wall at the side of the blood vessel, and they have a cell-cell junction which is called a podosome, and that podosome is an important for adhesion, but also sort of for mechanical sensing of the surroundings. So these monocytes they are circulating in blood. They are rolling across the surface. They have contacts with selectins, and when they're rolling across the surface of the endothelium, if they detect some chemokines, then they can be activated. And once they're activated, then they can form strong mechanical contacts. They then have to find the gaps between the cells, and then they squeeze out between the endothelial cells, and then they can go and give you an immune response and be active. And so the podosomes are involved in this strong mechanical contact, and also they probe the surface to see how hard it is in order to be able to tell where the gaps between the cells are. And so in the literature at the time, there was some ideas that when this podosome is formed, it's formed between the contact between an integrin LFA1 and with an ICAM, which is a cell adhesion molecule in the surface of the endothelial cells. And there was this idea that the LFA1s were pre-clustered, and so that there could be some preferential size in order to form this mechanical contact. So we did some experiments in a simplified form of this. So we pre-activated our monocytes using a chemical agent. We did it in a sort of static condition, so it's not sort of like the real situation. It allowed us to study the formation of these podosomes with this LFA1 ICAM interaction. And so here we did the same, that we took the ectodomain, the extracellular domain from the ICAM, it's fused to an FC domain, and then we can assemble it along the surface. We can create our different pattern sizes, and then we can interact. And so here is sort of a dotted line is showing roughly the size of one of these podosomes. It's 600-800 nanometers. And so we've got a few of these patterns underneath the place where one of these podosomes could form. And then what we see is that If the patterns are too small, we don't get any adhesion at all. So these are 100 nanometer patterns, and this 160, and that's the same as our sort of negative control with no protein, no patches. And then we have sort of intermediate adhesion level from 200 nanometers and up. And then we have sort of full strong adhesion when we get up to this 500 or 800 nanometers in size. And so these adhesions are, they're characterized by having sort of a punctated, bright core of actin, and then also staining for vinculin. And so these protozomes they have a particular structure to them, and that's because they have this role as being a mechanical sensor of the surroundings of the cell. And it can either be to look at the mechanical properties of the extracellular matrix or the cell membrane of neighboring cells. And so the formation, these protozomes are the way they're understood is that they have a ring of integrins. These are integrins in the surface of the cell. And this example here is from the literature, but this is for binding to extracellular matrix, but it's the same for the cell-cell contacts. So you have this ring of integrins, and then in the core there is actin, and that the actin is being pushed out through the cell membrane, and in that way it can probe the surroundings and feel the force. And so it has this formation where it has actin in the middle, and then it has a ring of integrins, and then also have of the vinculin that will form to adapt to protein. So we can stain for those proteins, and it's getting slow again. So we can stain for the actin and vinculin, and we can see how the protozomes that form on our nanopattern surfaces, how they look. And so for the very smallest patterns, we were getting no adhesion at all. So obviously you can't study much there. But then for the intermediate patterns, I'm now waiting for my slide to change. For the intermediate patterns, there are several of these small patches can interact with a protosome or start for the formation of a protosome. It would really help if I had the pictures. You'll have to bear with me for just a second.
Should be fine.
So for the smallest pattern sizes up to 160 nanometers, and this red line is meant to be roughly the size of the protozomes that we see when we see them. So here there are definitely several patches available, and wherever the cell tries to form one of these protozomes, it'll find patches of this ICAM available. And so what we believe is that each individual patch has to be a certain number of integrins involved, and we sort of come up with a number of about 9 of these integrins have to be involved before it can start to form an adhesion. And then we have this intermediate stage where these are images from the cell surface. We're looking at the actin and looking at vinculin. The vinculin forms small regions which fit with our patch size, so we're able to control that. And then it's sort of forming like a triangle or however many it can fit around one of these protozomes. And it seems to be sort of getting intermediate contact, but not strong contact. And then once the patch gets big enough, then we can actually, for the 800 nanometers, we can fit a whole protosome on top of one of these patches. And so we again see our core of actin in the middle, and then the vinculin around the outside. So we come up with this idea that there's 2 things involved. One is that there needs to be at least 9 LFAs in order to form, none of these LFA1s in order integrins to be able to form an adhesion. And then once you have an adhesion, it sort of matters how many there are. And yes, okay. So this vinculin has this protosome has a ring shape to it. So we actually were interested if we took our 800 nanometer patch, which was covered with ICAM1, if we just take out the core of that, in principle, the core is not important, would it change our adhesion? And so actually what we see is that the cells are quite happy to adhere. There's a load of different structures here. But these are these circles, and these are rings where we've either had 800 nanometer rings or 500 nanometer rings, and then we've sort of varied the wall thickness of the ring. So if you want more details, you can go and look at the paper. In the end, actually what we find is that if we make rings, then the cells will happily stick, but they will not always stick in the middle of a ring. And so what we find is that it actually only needs to have 2 contact points in order to be able to form a podosome. So this whole ring structure is not fully needed in order to get the assembly of this and the adhesion of the cell. So that was an example of a cell-cell contact between monocytes and endothelial cells. So I've given a couple of examples of some work that we've done in the past. What I wanted to talk about now is to go back to the engineering. And I say that because all of this is driven by having the ability to pattern proteins and to control where they are placed on the surface. And so in the last couple of years, we've been sort of working on some challenges to that. And so one challenge is that I have 3 listed here. There are more, but these are ones which I think are important. So having a pattern which contains just one protein it has some interest, but it really limits the type of questions you can ask about the cells which are adhering. A second thing is that it's always a problem for us is that non-specific binding of proteins will allow the cell to change its environment. I mean, it's constantly producing protein, releasing it, and remodeling its surroundings. And so we've been working hard to reduce that non-specific interaction. And then the last is that when we make a pattern like this, if we have a sort of a 200 nanometer patch, we sort of imagine that we might have some tens of proteins in there, but we don't control where they are. So it would be good to go to a higher level of resolution to get to the molecular scale. So these are 3 things that we are actively working on. So I'll take the first, which is this idea of having more than one protein in a useful way, I'm saying here. And so if I go back to this sort of monotype experiment that we were doing, and so here, in the real situation, monocytes which are not active are interacting with selectins, and then they're rolling along the surface in flow. They can be then activated by interacting with another protein, a chemokine, and then they're forming an adhesion contact with actually a third protein. And so to study this, really, you would want to have this having several proteins present on the surface in a controlled fashion, and you'd also want the whole thing to go on in flow. So that's something that we've worked a little bit on. Here, we could work with, we're working with the same platform of having a sort of gold and silicon dioxide chemistry. We covered the silicon dioxide with a protein rejecting chemistry, but we allow then coupling chemistry between those polymers to allow us to couple something to the surface. So that's kind of the easiest way to have a protein nanopattern with 2 proteins, is to have a nanopattern of one and then just fill the background with another. And so in this work, we were working with E-selectin and P-selectin. So this E-selectin and P-selectin are relevant because the expression of those E and P selectins changes when there's an inflammatory situation. And so that's one of the signals that a monocyte can use to tell when it should stop and leave the blood vessel. And so using this system with his tags, we were able to pattern either side of a fluidic channel and then have this sort of 2-way patterning going on. And then just to show that it matters, so this is where we have made movies of monocytes in the microfluidic channels. The right side of the channel has both ICAM nanopatterns and E-selectin in the background, and the left channel has just the ICAM. And with just the ICAM, we don't get any adhesion at all, basically, in flow. And so in the real situation, you need to have this rolling along the surface in order to give this contact. And then this is showing the rolling events. And so at least having the 2-way patterning, you have a possibility to bring the cells into contact with the ICAM. Yeah. So that was the idea of just sort of putting another protein in the background, and that works. There are times when you would want to have several proteins nanopatterned so that you don't just have them in the background, but you have a control of where they're placed on a nanometer scale. And so this is work from another graduate student in the group, Ali Sharokhtash, and he's been working with making 2-way and 3-way patterns of proteins, and he's making different pattern sizes. And in this system, it's a core, a disc, or a circular region of one protein with a ring of another protein around it. And so he's developed a system. It's a slightly more complicated process from the earlier system, but a good thing with it is it can work on glass. So what he does is he works with a polymer resist. We use colloidal lithography to create a whole mask, which has sort of, here it's a hard mask with a hole in it. and then he can deposit through that a chemistry, like, for example, titanium dioxide or chromium, to change the chemistry in one part of the surface. And then using this double layer resist, he can remove the first layer of resist, and then he then has etched out a region here around this disk, which he can then functionalize with a PEG which contains some functional group on the end. And so this is a PLLT PEG which contains a biotin group, for example. He can then lift off the second polymer and then backfill with some PEG or other stealth polymer in order to prevent protein binding here. And then you can sequentially bring in one protein which will non-specifically just adsorb to the center part, and then you can have coupling through the biotin of something into this ring. And so this is what's shown up here. And so it seems to work quite robustly. It's not something that we've yet used on cells, so we're now working out what's an appropriate molecular biology question that we can ask of a cell using that system. So then I wanted to comment about this non-specific protein binding being a problem. So our previous sort of platform was this idea of having a gold surface which was made hydrophobic, and then having silicon dioxide where we stop proteins from binding. And then we just allow proteins to non-specifically bind on this hydrophobic surface. And it actually works pretty well for extracellular matrix proteins, but it has a problem. And that problem is that you don't prevent all background binding here. And so if you bring in, serum, or if you have cells which are present there for long enough, then they produce their own matrix and remodel, and you can have binding of some of these proteins within this patch. And so if you're trying to look at cell-cell contacts, a major problem is that you have extracellular matrix proteins bind here, and then the cells bind through focal adhesions. And so, for example, in the studies we did with ecadherin, we couldn't work in serum because we would always just get focal adhesions forming. And so you can do studies, that you can design good studies, but it's severely limiting particularly if you want to look at long-term culture, or if you want to look at functional effects of cells. You really want to have the ability to have different media agents added. So Ali has worked on that too. He has come up with this using the platform, like I mentioned before, but there he can have a complete stealth background, so that there is sort of PEG or other protein rejecting polymers which are then functionalized at the top. You can either functionalize it just in a patch or in the background, and then you have the benefits of the PEG, but still have these protein nanopatterns. Yeah, so that's something which is working now, and it's something which actually Sadegh has been applying that to look at another type of cell-cell junction. And so this is the desmosomal desmosome junction, which is like the hemidesmosome. It connects the outside of the cell to the intermediate filaments, so the keratins in the epidermal stem cells, but here it's a cell-cell contact. And so this desmosome is characterized in the TEM by these sort of dark, intensely staining regions which are dense in protein. And in that junction, you have different proteins, types of proteins, desmoglines and desmocollins, who form this. And then they have adapter proteins that will connect that to the intermediate filaments, which you see here. And so this is something which is present in skin. What's actually interesting is that these desmoglein and desmocollin proteins are differently expressed at different layers of the skin, which actually corresponds to different differentiation states of the keratinocytes. And so for us, it's an interesting thing to look at whether this affects the differentiation of the cells. And now it's being slow again. So Sadegh has been taking one of these proteins which are present inside of the desmosomes, DSG2, and forming patterns of that on a surface, and then seeing whether he can assemble desmosomes from stem cells, epidermal stem cells on top of that. And the pictures look cool, and you'll see them in just a minute. I hope. Yeah, so I'll start telling you the outcome. So if we'll see that there's a pattern of DSG2, which is the proteins that he's placing on the surface, and then on top of that, when you put the cells on top, you can stain for DSG2, and you find DSG2 everywhere because we've put it there. But then you can see a larger amount of it because you have the cell producing DSG2, having it in the membrane and forming it into the patches. And again, it's forming controlled by the size of the patches. But then he also stains and can see the desmocollin, DSC2, is being assembled by the protein cells as well. And so it really feels like we're having this assembly of the desmosome, and we're able to mimic it on a pattern surface. And so the next step after that will be to see whether this is involved in this cells at the basal membrane committing to, so here's the pattern of the DSC2 on the surface. Here are the cells, and you can see the pattern in the background. But then you have a higher intensity of this DSG2 because it's the cell protein as well. And then this is the DSC2, and so we're getting both of the proteins forming, which will indicate for us that we're forming this desmosomes. And as the next stage will be to see how that's involved. Is there a role for the desmosomes in cell signaling and decision to differentiate? So the last thing I just want to talk about is this sort of a look to the future. So we are conscious that we don't control the proteins on the molecular scale. And so within this patch that we form, we don't control where these are positioned. And so that's actually the this current center that we have, this CellPat, Center for Cellular Signal Patterns, where we are using DNA origami, which can then control placement of proteins on the scale of, perhaps 4 nanometers up to 10 nanometers. And the idea will be to integrate this with this nanoscale patterning in order to have a control of where the ligands for cell binding are. And so one of the central things in this project is to look at multivalency effects and avidity. Okay, so I've given some different examples. The main take-home message is that by using nanoscale protein patterns, you can actually control the formation of a whole range of different cellular adhesions. And so that can either be a tool for studying cell function or the formation of adhesions, sort of molecular cell biology. But it can also be a tool for an engineering tool for creating materials or tissue engineering products or things for doing advanced cell culture in order to control the cells which are in contact with that surface and perhaps giving them a better sort of physiological phenotype. Before I stop, I should thank the people that did the work and the people that paid for it. So there's a whole bunch of different funding bodies, and in particular, the Max Grundforskning Fund, who are funding this CellPat Center. There are former group members, and then there's Ali and Sadegh, who are, you've seen this picture before. Now I can point out that's Ali and that's Sadegh. It was taken before Corona, so hopefully soon we'll be able to do the same again. But actually, the next event that we would have gone to has already been cancelled and pushed back by another month. So we'll have to wait and see. And then also I should thank and acknowledge the international collaborators on these projects. And I'll thank you all for your attention.
Great. Thank you so much, Duncan, for your nice presentation. It was great talk. So let's get started with the discussion section.
Should I stop sharing?
Yeah, that would be great. So if anyone has any question, comment, suggestion, please turn on your camera or microphone or write it down in the chat box. I can answer the question. Nothing.
So can I have your email?